So our next topic in carbohydrate chemistry is classification of carbohydrates, in which we'll be doing monosaccharides, disaccharides, oligosaccharides and polysaccharides. Carbohydrates are known as saccharides also, that's why the name is monosaccharide, like that. Okay. Now mono means 1, di means 2, oligo means 3 to 10 here, and poly means more than 10. What do you mean by when I say monosaccharide has 1? 1 what? Monosaccharide has 1 what? Is it 1 carbon? No, because glucose, glyceraldehyde, fructose, galactose, they are all monosaccharides, which we have done in isomerism, right? Now, disaccharide will be like 2 glucose joined together, example maltose, that will be a disaccharide. So this number is what? This number is not number of carbons, this number is number of carbohydrate unit. This is number of carbohydrate unit, so be careful here. This is not number of carbons. So we'll be taking them one by one. First we are going to take is monosaccharides. Now number of carbons in monosaccharide. Number of carbons in monosaccharides range from 3 to 9. That is minimum carbon is 3, maximum is 9, and there is no carbon with there's no carbohydrate with 8 carbons. For example, 3 carbon aldehyde is glyceraldehyde, 3 carbon keto is dihydroxyacetone. This we're already done. 4 carbon aldehyde form is erythrose. Keto form is erythrulose. 5 carbon aldehyde form is ribose. Keto form is ribulose. Then 6 carbon also you know, glucose and fructose, right? Then in case of 7 carbon, it is glucoheptose, and keto is pseudoheptulose. Then there is no carbohydrate with 8 carbons, so we don't have any example here. In case of 9 carbon, there is a modified 9 carbon aldehyde sugar known as sialic acid or N-acetyl neuraminic acid, but there is no keto form of 9 carbon carbohydrate, right? So no example again here. Now next is we have to do the reactions of monosaccharides. So write down reactions of monosaccharides. The first one is I'm going to take is oxidation. By oxidation, all the monosaccharides, they will form acids. By oxidation, they form acids. If oxidation occurs at C1, then aldonic acid is formed. If oxidation occurs at C6, then uronic acid is formed. If oxidation occurs at both C1 and C6, then saccharic acid is formed. Saccharic acid is formed. In case of glucose, the name of acids will be, in case of glucose, it is gluconic acid here. It is glucuronic acid here. And in case of glucose, saccharic acid is the name when it is oxidation at both C1 and C6. Now, if they ask you oxidation at primary alcohol of a carbohydrate will give rise to which acid? See, primary alcohol is same like C6, so you will mark uronic acid here. But let's see which is primary, secondary and tertiary alcohol. Let's say I have this carbon skeleton. This carbon skeleton is there. If OH present here, it is primary. If OH present here, it is secondary. And OH present here is tertiary. What do you mean by this? See, this OH is attached to a carbon which is further attached to one carbon, so primary. This OH is attached to a carbon which is further attached to 2 carbons, so secondary. This OH is attached to a carbon which is further attached to 3 carbons, so it is tertiary. Now let's see in case of glucose, which is primary alcohol. In case of glucose, C6 is CH2OH, which is attached to C5. C5 is further attached to C4, right? Now this OH of C6 is attached to a carbon which is further attached only to one carbon, that is C6 is attached to C5. So this is primary alcohol. Therefore, in case of carbohydrates, if they say oxidation at primary alcohol, then you will consider C6 because prototype of all the carbohydrates is glucose, and now you will mark uronic acid, right? Now see the second reactions of monosaccharides which we have to do is next is reduction. So I told you that by oxidation monosaccharides form acids. Now by reduction they all form alcohol, and all these alcohols are hygroscopic in nature. Hygroscopic means they will attract water, they will absorb water. So can I say if this kind of alcohol is present in excess in some cell, then it will cause cell swelling, right? So let's take the example. Example is glucose. Glucose gets converted to its alcohol known as sorbitol. Sorbitol is also known as polyol. Then galactose is converted to its alcohol known as galactitol. Galactitol is also known as dulcitol. Then mannose is converted to its alcohol mannitol, right? Now let's see the clinical point here. Sorbitol, when gets accumulated in body, it especially gets accumulated in lens of the eye, causing cataract. That is, it will cause snowflake cataract. On the other hand, galactitol will cause oil drop cataract. Galactitol will cause oil drop cataract. Now, therefore, I can say that in diabetic patient, snowflake cataract will occur. Although cataract is a rare thing in diabetes patient, but if it occurs, it is of which type? Snowflake cataract, right? And in galactosemia patients, it is oil drop cataract which will occur. And the use of mannitol, you know that mannitol is used to reduce intracranial tension, again because of its hygroscopic nature, because it will attract water also. So it will help in the reduction of intracranial tension. You can once see the picture of oil drop cataract and snowflake cataract. In case image-based question comes, then you should be able to recognize these structures. Now coming to disaccharides. Disaccharides are made up of two monosaccharides. Let's say this circle is a monosaccharide. Disaccharide is two monosaccharide joined together with a bond, and that bond is known as glycosidic bond. For each disaccharide, you have to learn the glycosidic bond. So let's take example. First is maltose. Maltose is made up of two glucose. Next is isomaltose. Isomaltose is also made up of two glucose. Next is trehalose. Trehalose also made up of two glucose molecules. So I have taken these first three compounds which all contain two glucose molecules. But the bond is different that you have to learn. In maltose, it is alpha 1 to 4 bond. In isomaltose, it is alpha 1 to 6 bond. And in trehalose, it is alpha 1 to 1 bond, right? And maltose is reducing. Isomaltose is also reducing sugar, but trehalose is non-reducing sugar. Next example I'm going to take is sucrose. Sucrose is made up of glucose and fructose, and the bond is alpha 1 to 2, and sucrose is also non-reducing. So trehalose, sucrose, both are non-reducing. And the last example I'm going to take is lactose. Lactose is made up of galactose and glucose, and the bond is beta 1 to 4. And note here, this is beta bond in lactose. And in rest all the disaccharides, I have told you alpha bond. This is beta bond in lactose. And this is reducing. Lactose is reducing. So only two non-reducing we have done, that is trehalose and sucrose, right? So we have seen which disaccharide is reducing, which is non-reducing. But what about monosaccharides? Monosaccharides, all monosaccharides are reducing, right? Monosaccharide functional group is always free. So all the monosaccharides are reducing. Reducing means functional group is free, right? If functional group is free, then the sugar is reducing. If functional group is not free, then the sugar is non-reducing, right? Now, let's see some tests given by monosaccharides and disaccharides. First is, I'm going to take is Molish test. Molish test is a general test which is given by all the carbohydrates. But there's one condition, that number of carbons should be 5 or more. Only then Molish test will be positive, right? Next is Benedict's test. Benedict's test is given by reducing sugars. So can I say all monosaccharides will give Benedict's positive? Because all monosaccharides are reducing, right? Then the third is Selivanov's test. Selivanov's test will distinguish between keto sugar and aldehyde sugar, because Selivanov's test is only given positive by keto sugar. So if Selivanov's positive, then we are very sure it is keto sugar. Now one more test here, that is Barfoed's test. Barfoed's test is again used to distinguish two things, that is it can distinguish monosaccharides and disaccharides, and it is given positive by monosaccharides. So if Barfoed is positive, we are very sure it is monosaccharide. If Selivanov's is positive, we are very sure it is keto sugar, right? Now routinely blood glucose estimation is done. At that time, which test is used in the lab for doing this blood glucose estimation? That is one enzymatic method known as GOD-POD test. GOD-POD test is enzymatic test. See the full form. GOD stands for glucose oxidase and POD stands for peroxidase. Any enzymatic test is very accurate test because enzyme is very specific for its substrate, right? And this test is a routinely used test which is done. This is a very cheap test which can be easily done in just 30-40 rupees. Any lab can measure blood glucose levels of a patient, right? And in this test, what happens? When glucose is there in the sample, it is first taken up by GOD enzyme, and now oxidation will occur at C1. You know, when oxidation occurs at C1, then aldonic acid is formed. In case of glucose, it is known as gluconic acid, and plus some amount of H2O2 is also produced, that is hydrogen peroxide. Now this hydrogen peroxide will be taken up by peroxidase enzyme and some colored compound is formed. You know, in biochemistry, we have principle of colorimetry. If some colored compound is formed, then we can estimate the amount of that analyte which is used in that reaction where colored compound is formed, right? So easily that can be measured. This enzymatic method, because it is enzymatic method, that's why it gives very accurate results, again because enzyme is very specific for its substrate, right? Okay, now after disaccharides, our next topic is oligosaccharides, but we will not do this topic because oligosaccharides do not exist as such in nature. They are just breakdown product of polysaccharides. Now directly coming to polysaccharides. Polysaccharides are of two types, homopolysaccharides and heteropolysaccharides. Homo means same, they are made up of same carbohydrate units. Hetero means different, they are made up of different carbohydrate units. Now just think once that heteropolysaccharides are branched or unbranched. What do you think? Hetero are actually unbranched. It appears as if they are branched, they are complex, but no. Heteropolysaccharides are unbranched. In fact, most of the homopolysaccharides are branched. So first we'll be taking homopolysaccharides, in which the first two example I'm going to take is starch and glycogen. So first is starch. You know, starch is present in plants. It is the storage form of carbohydrate in plants. And the second one is glycogen. Glycogen is present in animals, right? Now, the starch is less branched, but glycogen is more branched, because the branching point in starch comes after 24 to 30 glucose residues, but the branching point in glycogen comes after 8 to 12 glucose residues. That means, let's say this is a branch in starch, then 24 to 30 glucose residues, then next branch comes. Then again 24 to 30 glucose residues, then next branch will come. But in case of glycogen, when there's a branch, just 8 to 12 glucose residues, again branch, 8 to 12, again branch. So glycogen is more branched than starch. So these were the differences in starch and glycogen. But now, what are the similarities in starch and glycogen? The similarities in starch and glycogen is that both are made up of alpha glucose. Both are having alpha-1 4 bonds in the straight chains and alpha-1 6 bonds in the branch chains. So let's say this is a glucose residues. This is a glucose residue and all these glucose residues are joined in a straight chain. Now here I'm going to make a branch chain. So tell me the bonds. What is the bond here in the straight chain? This in the straight chain, this one, all in the straight chain are alpha 1,4 bonds, but all these of branch chain are not alpha 1,4. Only this, the first bond at the branch point is alpha 1,6 bond. Rest again all glucose residues. See, this is a branch chain in this structure, but see, all these glucose residues are again arranged in a linear sequence. So all these bonds will be alpha 1,4 bonds. So to be very precise, we should say that only the branch point is alpha 1,6. Only the branch point, one point, right? This point, only the branch point is alpha 1,6, not the full branch is alpha 1,6, right? Now, starch is made up of amylose and amylopectin. Starch made up of amylose and amylopectin. This amylose portion is 20% portion and this is the unbranched portion of starch. But amylopectin is 80% and this is the branched form of starch. Now tell me one thing, that if I have to compare glycogen with one form of starch, then I will take which one? Amylose or amylopectin? Answer is obviously amylopectin, the one which is branched, that is taken for the comparison with glycogen. I will use this point in glycogen storage diseases. So remember this point, right? Now let's see digestion of starch. In digestion of starch, you know, starch is a plant product coming in our diet. So when starch is coming in diet, it will go to intestine. First, it will be broken down by which enzyme? Amylase. It is not starches. It is amylase because starch is made up of amylose and amylopectin. That's why the name taken amylase, right? Now, amylase breaks which bond? Alpha 1,4, alpha 1,6 or both? This is a frequently asked question. Amylase breaks which bond? And let me tell you, amylase breaks alpha 1,4 bond. So when amylase breaks alpha 1,4 bond, then here a structure is left which is having lots of alpha 1,6 bond known as limit dextrins. This is having lots of alpha 1,6 bonds known as limit dextrin. Now further limit dextrin will be taken up by some enzyme which is now going to break down alpha 1,6 bonds, and that enzyme is isomaltase. I told you isomaltose molecule, isomaltose disaccharide has 2-glucose and alpha 1,6 bond. So isomaltase enzyme will break down these alpha 1,6 bonds, and now by the combined action of amylase and isomaltase, we'll be getting lots of disaccharides maltose. Now these disaccharides maltose will be taken by enzyme maltase, which will break down the remaining alpha 1,4 bonds, and in the end we are getting our monosaccharide glucose, which is absorbed from the intestine. So this way starch is getting broken down in the intestine, and the first enzyme is amylase. You know, we have salivary amylase and pancreatic amylase, right, which breaks alpha 1,4 bond only. So note here that this polysaccharide starch is first broken down to oligosaccharide, then disaccharide, and then monosaccharide glucose we get, which is absorbed from the intestine. This digestion of starch is very important. Now after starch and glycogen, the next homopolysaccharide we are going to take is the third one, dextran. Dextran is a homopolysaccharide which is again made up of alpha 1,4 and alpha 1,6 bonds, made up of glucose only, alpha glucose, but it is also having alpha 1,2 and alpha 1,3 bonds. So this kind of structure is high molecular weight structure, highly branched structure. This kind of molecule is not broken in human body and it is having high molecular weight. So therefore this dextran, see, intravenous dextran is used. If I give intravenous dextran, it will not be broken down, right? And it is a high molecular weight compound. It will stay in the intravascular fluid. So this IV dextran is used as plasma volume expander in patients of hypovolemic shock. Plasma volume expander in patients of hypovolemic shock. IV dextran used. And you know, we have a technique chromatography, gel filtration chromatography. In gel filtration chromatography, the gel which is used is biochemically it is dextran which is used here, and also the dental plaques which gets deposited the dental plaques which gets deposited in our teeth, they are also network of dextrans, right? Now the fourth homopolysaccharide we are going to take is cellulose. Cellulose is unbranched. I told you most of the homopolysaccharides are branched, but cellulose is unbranched, and cellulose is made up of beta glucose. Let me tell you some general point about beta. Whenever there is beta bond in any molecule, I will tell you a general point. Beta means not broken. Beta bond is actually very difficult to be broken. So it can't be broken by our enzymes. We don't have any enzyme to break beta bond. So this is a general point that beta is not broken. And when beta of cellulose is not broken, then cellulose act as fiber in the diet. That's why cellulose act as fiber in the diet. We have two type of fibers, insoluble fibers and soluble fibers. Example of insoluble fiber is cellulose, hemicellulose. An example of soluble fiber is pectins, gums. Now, what is the difference between soluble and insoluble fibers? These insoluble fibers, they are excreted unchanged from the intestine. That is, in its original form, it is thrown out of body. But soluble fiber, they will attract water. They get converted to gel form. They attract water, gets converted to gel form, and then this gel is excreted out of the body. So can I say soluble fiber is better in preventing constipation? Because when soluble fiber is present in the intestine, it will attract water also. So it will give more osmotic load to the intestine. So therefore, soluble fibers are better than insoluble fibers. Now, fifth homopolysaccharide is inulin. Inulin is made up of beta fructose. Again, there is beta bond. So inulin, if ingested, cannot be broken down, right? And uses of inulin are, it is ideal substance for the measurement of glomerular filtration rate, and also inulin acts as prebiotic. I have not written probiotic, I have written prebiotic. Understand the difference. Probiotic is bacteria, live bacteria we are giving, that is probiotic, right? But prebiotic is food for the bacteria, like inulin, right? Inulin, if we ingest, it is not broken down by our enzymes. It is beta bond, right? So from small intestine, it is not broken down. So it will go to large intestine. In large intestine, bacteria are there. They can break this beta bond and they can use this carbohydrate fructose as their food, right? These days, whenever good prebiotic capsules you can see, it is written prebiotic plus probiotic, both are given together, right? So we are done with homopolysaccharides. Now next coming to heteropolysaccharides. Heteropolysaccharides are a little difficult to understand. So I'll try to make it very easy for you. First, let's understand the names. There are 2-3 names, heteropolysaccharides. So let's see. First name is hetero. Why the name hetero? I've already told. Because it is having different carbohydrate units. And also the name is, they're also called GAGs. GAGs is glycosaminoglycans. Glycosa means carbohydrate, but amino. See here, amino word written here. That is a very special point. See here. We have carbon, hydrogen, oxygen. These 3 atoms are present in carbohydrates. And we also have carbon, hydrogen, oxygen in fats or lipids also. Only these 3 atoms are mainly present. But in case of amino acids and proteins, in addition to carbon, hydrogen, oxygen, nitrogen or amino group is present in case of amino acids or proteins. So amino group is a specialty of amino acids, and carbohydrates specialty is OH. We did polyhydroxy. So OH is the specialty of carbohydrates. Amino is the specialty of amino acids. Now, can you see this GAGs is having amino group? So this is a very special carbohydrate which is having amino group. Otherwise, carbohydrates don't have amino group. But this is a very special carbohydrate which is having amino group. Now, one more name, they are known as mucopolysaccharides because they are found in mucus secretions, because they have a role in lubrication, cushioning effect. So, mucus secretions. Now, definition of gags, I'm going to write a very simple definition. I'm going to write tandem repeats. They are tandem repeats of two different things because they are heteropolysaccharide, two different things. And now they are tandem repeats of ASUA. I have written the short form, full form I'll be telling later. But first understand tandem repeats. What do you mean by tandem repeats? One thing is repeated again and again. That is tandem repeat. Repeated one after the another. Now ASUA is repeated again and again, one after the another, like this. ASUA. So this is the structure, a very simplified structure I have made. And note here, I have not made any branch. I told you heteropolysaccharides are unbranched, right? So I have not made any branch here. Now see the full form. AS stands for amino sugar. See, there are lots of amino group here, amino sugar. And UA stands for uronic acid. You know that C6, oxidation at C6 makes uronic acid. So they are tandem repeats of amino sugar and uronic acid repeated again and again. And all the gags, they combine with proteins. All the gags combined with proteins to make proteoglycans. So they're also called proteoglycans. Let's see the difference between proteoglycan and glycoprotein. Always the part which is written later, that is protein here and glycan here, that is more. Glycan means carbohydrate. So in proteoglycan, carbohydrate is more than protein, but in glycoprotein, protein is more than carbohydrate. Now in case of proteoglycan, the example we have done gags. Gags is the example here. And the carbohydrate of proteoglycan is always a heteropolysaccharide. We have done that. But in case of glycoprotein, example is collagen. The fibrous protein collagen is a glycoprotein, means mainly it is a protein, but some portion of carbohydrate attached to it. You know, plasma proteins, there are many proteins present in plasma. All the plasma proteins are glycoproteins except albumin, because albumin is not having any carbohydrate attached to it. Albumin is only protein, right? Now, carbohydrate of glycoprotein is either a monosaccharide or oligosaccharide. It is never a polysaccharide. So this is the difference between proteoglycan and glycoprotein. You have to be very careful with what is written in the question. Both these are made up of carbohydrates and proteins, but there are differences. Now these gags, glycosaminoglycans, they are highly sulfated because you can see that from their examples. Example of gags are heparin sulfate, chondroitin sulfate, keratin sulfate, sulfate. Every time sulfate is there in the name because they are highly sulfated. You know, they are unbranched and they have lots of acid carboxy group. So they have sulfate, sulfate gives negative charge. They have carboxy, carboxy also gives negative charge, and they also have acetyl group. Acetate also they have. So there's too much of negative charge in the structure. Because of this negative charge, see my hands, there's too much of negative charge in the structure. This negative charge will repel each other, giving them slimy and slippery nature. So because of negative charge, they are slimy and slippery. And this negative charge is coming from sulfate, carboxy and acetate group. And because of slimy and slippery nature, therefore they are present in mucous secretions and known as mucopolysaccharides. Now there are two gags, that is heparin and heparin sulfate. Heparin and heparin sulfate. Heparin appears as if heparin sulfate is more sulfated. But no, the name is a misnomer here. Again, the name is a misnomer here. Heparin is more sulfated than heparin sulfate. Heparin more sulfated than heparin sulfate. And this heparin has highest negative charge because of this sulfate. Highest negative charge in which gag? It is heparin. And you know, heparin has a role in anticoagulation. How this negative charge has a role in anticoagulation? Because see, for coagulation, we need the positive charge of calcium. But now this too much negative charge of heparin will chelate. It will block all the positive charge of calcium, leading to anticoagulation. Charges are very important in biochemistry because direct questions can also be asked on charges, and when you understand charges, then it is very easy to understand so many concepts out of it. Okay. Now let's see the examples of mucopolysaccharides. Number 1 is hyaluronic acid. Hyaluronic acid is not having any sulfate. I told you all the GAGs are highly sulfated, but see the name hyaluronic acid. Hyaluronic acid, there's no sulfate in the name. So this hyaluronic acid is only GAG which is not sulfated. And also hyaluronic acid is the longest GAG, and the location of hyaluronic acid is synovial fluid and vitreous humor of the eye. Synovial fluid of joints and vitreous humor of eye, they have hyaluronic acid. The next one we are going to do is chondroitin sulfate. Chondroitin sulfate is present in cartilage. See, chondra means cartilage, so it is present in cartilage, bone, tendon, ligament, these kind of structures, right? Third is dermatan sulfate. Dermatan sulfate, as the name says, it is found in skin, also in blood vessels and valves. Then the fourth one is keratin sulfate. Keratin sulfate is not having any uronic acid. I told you all the GAGs, they have uronic acid, but which GAG does not contain uronic acid is keratin sulfate, a frequently asked question. And this keratin sulfate is present in cornea and connective tissue. It is responsible for the transparency of cornea. Then the next one is heparin. Heparin is having a role in anticoagulation, and it is released from mast cells and liver. And the last example of GAG here is heparin sulfate. Heparin sulfate is less sulfated than heparin, I already told you. And the location is, it is present on the cell surfaces. Location of heparin sulfate is very important. It is present on cell surfaces. Especially it has a role in retinal cell-cell attachment. Or if they ask you any cell-cell adhesion is caused by which GAG, then it is heparin sulfate, also role in retinal cell-cell attachment.